So thanks to many of you that submitted sort of comments on the form. There were a couple of groups that were clear outliers. So there was a bunch of people that were very interested in like data submission, data harmonization, data management, data everything. So I think that's a very natural breakout session. I propose that we just have it right here in this room. So that's sort of definitely topic number one. There was a second set of group that was very popular about talking about like a variety of tools and interoperability between AnVIL and other platforms and other systems. So that's great. What I propose there is we'll go right around the corner into the sort of the fireplace room, just through the lobby there. So that'll be a second group. There was a long tail of other suggestions that didn't sort of neatly fit into either of those two buckets. There are several of you interested in education, several of you interested in talking about other sort of phenotypes and other clinical data. So if you wanted to self-organize to the fireplace, just be aware that it might be sort of small numbers, or you're welcome to join kind of one of the two larger groups, either talking about data or tooling kind of next door. So those are the sort of the main topics that came up. Maybe we just do a kind of a quick show of hands. Like who here is interested in number one, data submission and harmonization? That's close to half. Okay, maybe even 2/3. Who here is interested in like tools and interoperability? That's like, I don't know, a third or so. And then do others like, I don't know, maybe you could raise your hand. I'm just kind of curious. Or maybe is everyone going to go to one of those two topics? Okay, so maybe I'd say in that case, let's try to focus it on these two topics. I think those are kind of the most sort of popular approaches. So we'll have our breakout now. In the slide deck, there'll be a few note takers in the group. What we're going to do is we're going to kind of come back at about 11 o'clock. We'll have a debriefing on how those breakout conversations went. That'll open up a nice transition into our town hall, which is just sort of a free form discussion. So for the next, I don't know, hour and a half or so, we'll just have lots of discussions about the state of the world for AnVIL. Okay, thanks everybody. Okay, so welcome. So this is the data room. So there's no kind of set agenda here. This is just sort of an open dialogue about the whole sort of data half of AnVIL. I don't know, does anyone have any sort of initial comments or thoughts? Some obvious things are, we've been hearing over and over again, there's a diverse set of data that are coming in from, a variety of different sequencing technologies. There's clinical data that we're trying to collect. There's lots of interest in single cell and spatial and imaging. So there's sort of a great diversity. There's a huge amount of it. Publicly released is something like 6 petabytes, but I think there's at least another 6 petabytes like in the queue. So there's kind of a huge volume of it. There's a variety of consent groups that can be, of course, important, but also silos the data and challenges. There's there's Duos and other platforms to try to help with that. I don't know. Do you want to kick us off here?
Well, so one thing I thought I liked. So one thing I appreciated that you had talking about DOIs for the workflows, right? I guess I just wonder, have we talked much about DOIs because we don't mint them in AnVIL for data sets either. Yeah, I stay here. Sorry.
Yeah, this came up yesterday when we were talking about potential features, new things for AnVIL. Oh, good. We have Jonathan. So others might want to say more, but it's definitely possible. It's not difficult for us to imagine that, for example, within, you could imagine DOIs for workspaces. We talked about this. There's a question, maybe Jonathan wants to say more, but there's a question about persistence. Because the thing about a DOI is, right, you don't really want that thing to change too much from under you, right? Like if you're creating one, there should be a notion that this thing is going to persist for some time because it's going to be part of the web of science, right? It's going to be cited for at least, ideally forever, but at least 10 years, let's say, right? I do think definitely, assuming that we're going to have data sets in AnVIL for at least 10 years, we could start adding DOIs to all the data sets, right? Like that would not be difficult for us to mint. We can obviously have our own unique IDs. That's not a problem. It's not, there's no harm in having those extra identifiers for our own purposes. But I think it would be sensible for us to just have that on our roadmap, like so that you can go, one value is if I want to, I need to cite the whatever latest HPLC thing, I'll just go to the Data Explorer, search for it, and there's the DOI, and now I'm just going to stick it in my paper. Like totally. That should be a thing.
Yeah, I think so too. And I guess I'm going to put Chris on the spot now too a little bit. I know that NIH has some agreements through things like DataCite Consortium and some of that. We had another project we worked with NHLBI to be able to get permission to mint IDs through them. And I just didn't know if it's something that NHRI has like works with DataCite or some of those groups, or if that's something we should consider. I'm also curious from other people, not us, the AnVIL leads too, if people think that's useful to have DOIs for AnVIL data, like I'm talking about submission here too.
NHGRI doesn't have anything specifically, but there are NIH-wide opportunities. I'm not remembering the most recent one I heard,
Okay.
but that's a thing. And yes, we should, happy to look into that. And I think it's available at, relatively easily.
Yeah. Okay. Yeah, great. I think, like I said, our experience with it was great. NHLBI, our program scientist was kind of like, and here you go. Great. All done. Is that simple? So
I think just to echo what Benedict was saying, there is a part of me that asks, kind of wants to play the antagonist and say, I don't think these are necessary. Why does anybody really want them? Is the answer because publications ultimately trust DOIs more than they trust our own UIDs? Then great, I'm persuaded. I think there's also a further question to say, are we doing this for data? That tends to be the first case. Are we doing it also for tools? And then from our discussion yesterday, should we think about workspaces as shared assets that have a DOI, that persistence in terms of time length, that I'm not, that's a hard one to estimate, but I think there's an idea of once a researcher has finished their analysis, they certainly don't intend to change that workspace. It may disappear, but it won't persist in a different form. So I think that's a different kind of persistence until oblivion. But I would ask about that as well. As one, just the thing we may want to promote more. I think that is a feature that the Terra infrastructure certainly has, maybe highly valuable. We've not really curated those, I think, as much as we could. They could be very helpful for outreach and kind of onboarding the next gen of researchers in AnVIL.
yeah,
I would ask if they need DOIs.
and that's specifically you have like this sort of publicly available workspace for people to view. Yeah, no, I think that's a great resource. Something all of us had adopted recently too, that I think has been really helpful, because you can just go push it out there. And I think in that the ecosystem is more lockdown. So letting people in and their data and tools, which you couldn't share otherwise, I think it's great. And I think that, one question about the sustainability, then, I think also we think about who owns that workspace. And right, is there a situation, because that's part of why it's hard. And once the data has been submitted, do you need those files in the buckets anymore? Like maybe there's ways that we could either make guidance so it's cheap to have that workspace persist, or some other way to, if we wanted to index those with DOIs, to like make them last, that they're sustainable. I don't know. There's just a thought too, but
I think it's a great idea having DOIs on the metadata around workspaces or projects or consortiums, with different levels. It feels like we have existing things now that could be repurposed to form those DOIs, specifically the DIRS IDs of actual files. The fact that a DIRS ID was minted gives some degree of formalization around that and intent for reuse. And I would say that I know it's emerging and I know it's incomplete, and I know caveat, but the snapshot IDs that are in the Teradata repository would be prime candidates for that. And the thing that piqued my interest in this conversation was looking for that carrot to support that effort to get things standardized, is of, hey, you won't get that DOI until it's in the Teradata repository and it's past QA and harmonization, normalization, all the boring things that never make it into a paper.
yeah. Did you want to chime in?
Yeah, yesterday we were also talking about I was talking about a database that we are interested in putting up on Terra and sharing. And where we went to for getting the DOI and everything was we went to Zenodo. And that was partly because everything by research showed that that's the one that's going to last the longest. But also, to be honest, when I was thinking about it last night, I remembered I went and looked at ColorsDB. And ColorsDB is hosted in Zenodo. I don't know if it's hosted in Terra, but I think it would be really cool to have it there as well. And so, yeah, I mean, that's just kind of how we chose, but I would really like to get it, de novo DB into Terra, and have people be able to actually query it in easy ways. I'm not sure how to do that yet, but we have You gave some great suggestions yesterday, though. Sorry.
I can respond specifically to that. So all of the compute was in AnVIL, and there is a Terra workspace that has everything. We also host a copy of kind of just the variant catalog in Zenodo. Part of that was just for a DOI that was easy to mint. I guess I'm interested, since you just went through this through DocStore, I've never like I've done this, I've minted DOIs using like Figshare type things, but I've never done it at the API level. Like, what is that process like? I assume like you don't load like raw data. It must be like some manifest that sort of describes what is present, and then that's what gets minted.
Yeah, I cannot comment on the actual API because I'm just a bean counter. However, so in DocStore previously, we had this rather like manual process that allows a lot of customization, but users had to basically click through to Zenodo, link their account, and then they could kind of they could then grant access through Zenodo to minting the DOI in DocStore. We decided that that was way too complicated, and that it was very clear that hardly anybody was actually bothering to do that. So yeah, we are just, there's a couple of things to mention. There is a cost associated with issuing a DOI, but it's not very large. And we did the math, and we'd have to be wildly successful for it. Like, it would be a great problem to have, right? For this to become, something that was cost prohibitive. It's like, it's not expensive. But yeah, you can become your own DOI provider, and there's like a whole mechanism that you can go through to do that. We decided that was absolutely not worth it. And so we are continuing to use Zenodo under the hood, and you can customize the whole bunch of stuff around the DOI so that it's clear that it's from DocStore. But they are ultimately the person that's the provider that is minting the DOI, and we just use their service. So yeah, there may be others within NIH that could also do that, and we definitely take that on advice. But yeah, it's not difficult.
Yeah, and I can comment a little bit. We've only tested the DataCite Consortium one, which again, there's a NIH partnership with them to do this. And so I don't think there's any cost to us as a repository for minting those DOIs. I think it's covered through some of these other agreements. Yeah, but I agree. I think it's nice because you have that we have a custom repository name in there, and it works pretty well. I think some of the biggest challenges that we're running into is we are collecting a lot of the information about what's the study name, who are the investigators, but there's, some of the rules you start to get, well, contributors versus authors versus some of these other terms that have really precise meanings. And you'd love to have ORC IDs, right? But I don't think we're capturing ORCIDs through AnVIL submission necessarily. And so some of these layers, we might want to shift a little bit what we collect, I think can empower them even further, especially when you talk about the Web of Science, when you start to really connect in, and now you can have your ORCIDs associated with the DOIs, and that citation, all that information flows really smoothly. So, but I think practically speaking, they don't make it hard. Like, they really don't. Once you get some of the precise mappings of what we're collecting as AnVIL repository and getting that shipped over to these groups, it's actually quite easy just to mint that. And then you need those sort of landing pages or your URL or your URI for tracking that, where they send you where they resolve the DOI for you. But I think that's something that we want to have anyway here. And once we've got that lined up in a couple of those pieces, it's actually very easy to mint the DOIs. So,
and just to add, so because we went through this whole process of like, sometimes a user, as you say, will want to customize the DOI and so forth. And so we continue to provide that even now that we are minting them. So we mint them on behalf of the user, but the authorship and all that stuff is described. And if somebody really cares and they do want to go in and add extra metadata, they actually have to go to Zenodo, but they can do that. And we've provided the documentation to do that. So fundamentally, we don't have to tie anybody up or make this that complicated.
okay. Yeah. Good to know.
I think just a comment too on the idea of sharing workspaces as, curated instances of analysis and data in the same location. I think we have sufficient storage so far, such as the data could rest within that workspace. It could also be pushed to TDR and then shared in that workspace later. It can also sit in the workspace on the AnVIL storage that we already have. So that there is a cost component as we think about bringing in maybe other ICs to this process and how they would kind of support that storage. We have different ideas we're working on for that to support them, but in general, I want to echo and report that, because a lot of people who are bringing data in are doing it post-analysis, and there's a good opportunity to get people going faster with building upon that by sharing both and not just the data.
Cool.
So I'm interested to hear from other people in the room, especially users. Are you able to find the data that you're looking for? Is it in a format that is accessible to you? You know, what would have been some challenges? Tyshell, I appreciate your comment about, you know, sometimes just the DB gap approval is the slowest part sometimes.
Yeah, once you have the approval, everything is pretty easy. I was like, just thinking, I was kind of hoping one thing that we've come up with in the lab recently is that all of the data that's in SRA, that's like old genome stuff, human genomes that aren't in AnVIL. So my bioinformatician is, there's a way you can actually click an SRA to have them go to the cloud like right away, which is super cool. But actually then it's a lot of time converting from SRA to FASTQ to, and you know, some of those legacy data sets that actually matter, and I don't know which ones those would be kind of cool if they existed as CRAMs in AnVIL, but I don't know if you ever want to go back. It's just like, not everything's in AnVIL, or Terra, I should say, but everything that is there is easy to access. I don't know if that's helpful.
Through the NCPI, this cloud platform interoperability, we do talk to some of the folks at SRA. You know, I think there's interest on both sides to make that process really efficient and easy. I guess one thing I would love to see, you know, we know on the protected access side, like GTEx is very popular, and TCGA and a few other cohorts are very popular. I don't have any idea of like what's popular in the SRA. I mean, it's such a complicated labyrinth of different data types and different quality and different history. So I don't know if there's there must be some place metrics on which of that subset is very popular. That to me, that would be a natural place to prioritize first.
Yeah, and I think Kavanik or Seven Ridges, I think actually did an exercise wrapping SRA in DERS or something like that too. I haven't used it myself, but you know, there's ways. I think it's very similar to that cloud bridge that you were describing, I think, but to kind of help make that available and index it in the systems.
We have been told that once we have RAS working properly, that should be fairly straightforward to allow access to including DB gap data. But I think that then means that Shell's question will become more pertinent of, are those the data people want? Is that, you know, are we then going to want to try to repackage some of that?
So just to kind of elevate the whole room. So RAS is this researcher authentication system, a single sign-on for NIH resources where you could sign into AnVIL and other cloud platforms. And in addition to single sign-on, it's a token where soon, hopefully, that token will say, yes, you are authorized to see these protected data sets. So it should really streamline that process of being able to navigate different platforms and getting access to different data. Yeah. Other questions, comments, thoughts about data harmonization? I don't know, Benedict, I appreciated in your presentation that it felt like, you know, most of the analysis was on 38 because that's where all the annotation is and that's where all the resources is. But, you know, you know, also showed quite convincingly there's a lot to be gained from looking into some of the new reference genomes. I've been thinking about, like, as you know, we've been thinking about like liftover. One thing I don't have a great handle on is like liftover into the pangenome itself. It seems like that could be a great solution because 38 is one of the haplotypes that's in there. CHM13 is one of the haplotypes in there, and plus much more. I'd be interested to get your impressions on like what we should be targeting for harmonization.
Yeah, no, thank you for that question. Definitely my wheelhouse. So we've been working on, so the algorithms to make it efficient to lift over between potentially thousands of references, because that's where we're going in terms of HPC. We've basically, they're fancy versions of BWTs, right? Sorry, this is to people who know what I'm talking about. We think we've got those worked out, and we definitely have liftover as one of the core things that the group will be deploying and providing. And so for example, the UCC browser provides a liftover tool, which is very popular. So it's like, but right now it supports 37, 38, and CHM13. We want to see it so that it's possible to do liftover between any specific reference. And obviously that has value, but actually in the long run, we see it, people increasingly, any human being is essentially a recombination of a small number of recombinations of of HPRC haplotypes, right? Modulo 10,000 small variants. That's it, right? And a few structural, very small number of structural variants. So we definitely see ultimately having okay, the thing you should be viewing is a personalized reference. And actually we can construct that, and you should want to like translate your annotations onto that structure. So in the long run, I think from the being very geeky about it, the computer science part of it is pretty well worked out. But obviously, like that's where nobody is really using personalized references and stuff yet. But I can totally see that is something that will happen over the next. It's definitely an application of the pangenome, and it's definitely something that the pangenome will make much easier. What I don't see as being super popular is, most of our haplotypes are, with the exception of maybe HD002 and NA12878 and a couple of benchmark samples, most of them are going to be just, they're just another sample in a very large crowd of samples. And so we don't imagine that people will be doing like, more than a handful of references. There probably isn't need to do specific liftover, but as I say, this like notion of being able to lift over to mosaics of haplotypes is definitely a thing that's going to happen, is happening.
Are you also thinking about how the other annotations may be impacted on different haplotypes? I mean, you just had a great example where I think it was a single SNP, change the splicing, and you get a new exon popping up. Like, how do we account for those sorts of changes?
Yeah, so GenCode is currently going through its renewal process, and as part of that, it is one of its major aims for the next 5 years, assuming it gets funded, so is population level annotation. So GenCode for the longest time has been like, we create the gene annotation, but it's the gene annotation of GRCH38, period, right? Which is great, but obviously does not capture all of the the many splice variants and so forth that exist in the population. So over the next 5 years, their plan is to take the kind of like automated output that comes from the HPRC in terms of just like annotation using standard machine approaches, and then curate that into a population annotation. And the idea is that what you'll have is, yes, a set of like canonical protein coding genes and non-coding genes, but then all of their like common haplotypic, not everything, but all of their common haplotypic variants that exist in the population. So that if you're doing like a single cell study, you can absolutely imagine including those structures in your analysis so that you can map and identify the existence of those in your study. So I totally see that's just yet another piece of how that kind of fits together. But it's going to take, it's not a super easy problem, as you can imagine.
Yeah, I mean, there's sort of arithmetic component.
Yeah.
But then, you get all these sort of subtle changes. Splice.ai, we've been playing a little bit with Splice.ai and with mixed results. Sometimes it gets it really strongly, and then other times it strongly disagrees with what the RNA-seq data shows. So yeah, I think that's an important research topic moving forward.
Yeah, just to mention, so we have generated connects for every single HPRC sample, albeit for the LCLs. These are LCLs, so we don't have it for every tissue and so forth. But the reason we generated that data was as a pilot, so we could go in and in this controlled setting ask, what is, for all of the genes that are expressed in LCLs, what is the sort of population level set of different transcripts and annotations? So we could kind of understand that for I think it's like 15,000 genes. And that should be super useful for understanding how important ancestry and diversity is in that level of annotation. And I think where we're going will be then in the future, when you've got a new sample and you have any information about it, you'll be able to essentially like figure out what personalized set of annotation you should be mapping to. And that should be much better.
Great. Other questions, thoughts about, I don't know, data searching, finding, people finding the data sets that they're needing, that they're looking for? I know this has become a popular topic about like which types of metadata do we really need to collect and expose and harmonize on? I don't know if anyone has any thoughts about this.
I want to just, because I don't want to speak all the time. So the current AnVIL data model, right, is pretty limited, and that's because it's hard to harmonize metadata, right? You've got to start somewhere. Obviously, Robert should talk about the clinical piece of this and adding information on that side, but you know, we were recently having some conversations about, well, what genome, you know, NHGRI is genome, right? For what Christ's sake, like we should want to have lots of genomic metadata, not to try and replace SRA or GenBank or whatever, but just make it super easy to find everything aligned to GRCH38 or everything that is at least 30x coverage or whatever, right? So I'm just curious, like what data fields would people really like to see that they currently can't, right? And we can, because the idea is we could prioritize these and then figure out how hard they are and how easy they are, and then, you know, move from there to start to try to add those.
I know what I would like to see, especially like when we go to SRA, is like, what is the depth of coverage of this genome? Like, what are the basic QCs? Because I hate spinning cycles, just running insert size metrics on short read data or fold coverage. You know, you can tell but from the file, like it's 1 gig. It's not a 30x genome, but I'm not sure how big, you know, how much X is there. And so like, I think that kind of metadata would be really helpful. And maybe there's only a small fraction of people that actually look at that. But, you know, if you have the QC, you can decide, is this a file I want to pursue any analysis on or not without having to get it and do it. Like that would be me in my nerdy ways.
That was precisely one of the topics that we've been discussing.
Yeah. How, hey, Brian, one thing. Yeah, I think a question for me is, this is super abstract. So do you trust like Benedict here to like come up with that list? Or how do you think we should get to a list, right? Because we've got a couple of names. At some point, we'll have to have, here's the official list of things we want, you should have, you could have. Do you want to like, if we had a community event and we said, hey, everyone, AnVIL's going to talk about what stuff you want, or do you just say, great, he gets to be the czar and whatever he says goes? I don't know how much you care about it, or like, what do you think the deltas are?
Well, I think Benedict is fabulous and he's made really
Yeah.
wonderful advances. So I trust that he has lots of really good insights on that. But I do think that you could actually have a call like NIH does calls for, you know, we're doing this step forward. I can't remember what they're actually called, but what is it? RFIs. Yeah, like requests for information. And then you could get feedback from the community. I think that those are super formal though. And I don't know, like people like me, I'm still early career and I don't know if I'm allowed to respond to those. I don't know. But you know, if it were a little less informal, then maybe
Yeah,
people would be.
I guess what I want to push on here is I think it would be awesome to have the community all come together and, hey, we sit down for an hour and everyone agrees and it's wonderful and sunny and the breeze is blowing, whatever.
No, I mean,
but,
like you come up with a list.
Yeah.
First.
Yeah. Okay.
A small group.
And then
of
we shop
people.
it out and get.
And then you send the list out. And then
Yeah.
if someone's like, oh my God, I want to see this other thing, then you think about if you want to add it. But like, I'm pretty sure you guys can come up with a top tier list first. And don't invite everybody to sit at the initial table. That will be a nightmare, I think.
Yeah, for sure. And I guess it's that question of like, would you be willing to contribute to this like consensus? And it sounds like the answer is yes. Okay. And that's what I want to get to. Because from where I sit, and I'm not a genome focused person, I would be like, sure, whatever. Yeah, Bennett, you got it. Like whatever you come up with, I'll abide by that. Sounds good to me. But yeah, that's helpful. So thank you. That's what I'm looking for.
Brian, did you have a comment?
I guess working up the chain from there, could we get the basic linkage? If I could just call that the assay information, working up the chain from there, could we get what specimens, and from specimen, what individual, and from what individual, what diagnosis or treatment or whatever? That would, I think that's definitely at 80%.
I will comment, how to put this politely. An extreme form of this, I think, has been, say, ENCODE, which has a very careful submission process of like every metric you could imagine, and a very detailed accounting of like how every byte of every file was generated. The upside to that now is there's a very well-recorded provenance of like where every drive file comes from, which sometimes that's really important. But I will say, having done this process in the past, like it's a high bar for submission and required us to go back and forth with the DCC like 100 times to like fix all these like little issues. So I think that, you know, the danger of having these really strong requirements is that people may just get like totally scared away, and it's only those groups that are really, really sort of mandated to submit will submit. So if we're going to have, I think, you know, these metrics are great and we need to have them whenever possible. We should strive for those that can be automatically determined, because then, you know, there'll be either, you know, a no bar or a very low bar to get into. So to me, it's all about, can we like automatically annotate as much as possible, or like maybe, I don't know, a wizard where we'll try to do as much automatically as possible. But if there's just a handful of fields that people need to fill in by hand, like that's a manageable list, as opposed to having like 10,000 characteristics that have to be populated.
Yeah, I think like a very small handful of fields, like if you look at the AnVIL Data Explorer that's there now.
Yeah.
Imagine if that was totally populated. And what the trade-off is between friction for submission, and then what do we do about the legacy submissions? Yeah.
Oh yeah, I totally agree with you. Like some of the things like the specimen, the researcher will have to tell you if it comes from blood or something else. But like the read coverage and QC, I think NHGRI should just give more money to Tara and have that as the first step, that when the file comes in, because I might do my fold coverage using QuickMare too, and it will give you a different estimate than Picard. So, or I don't even if you Picard got folded into GATK or whatever now, but essentially if you did it, if you had them have an automated, which you have workflows, right? So you could just have an automated workflow when the file comes in, then it comes back to the researcher and it says like, we ran this and it says your fold coverage is 30x, your insert size is this for short reads, whatever. And then they can look at it and just double check like, oh, that's good. And I don't think they're going to care at all. They're probably just going to approve it right away. And so maybe you can relieve some of the burden of them having to have all that information.
I like that idea of like, you know, it'd have to be a sort of a manageable cost, like, I don't know, $1 per sample, but like have you know, a bunch of metrics that are automatically computed for everything that is loaded. That seems like a really wise way to kind of improve the harmonization.
I was going to suggest something similar, so I like that idea. How strong of a guarantee does AnVIL have to make on the different information being presented like that? So, you know, nothing's perfect. Some of it won't be exactly right. Benedict showed on DocStore a way to tag information to say this was automatically generated. It hasn't been reviewed yet. Someone reviews it, you know, the tag goes away. And I like that idea on things. So where do we have to provide absolute guarantees, and where can we flag things and say, we're 95% sure insert size was this, but it actually hasn't been verified? Is that still helpful? Or are people going to say, or whatever, or are people going to say, no, 95%, I'm doing it myself? That doesn't help me.
I think like me, I would consider myself an advanced genomic researcher, but the team of people that you have at AnVIL and Tara are like top tier. Like, so if they calculated it and there's a way I can look in the DocStore at the workflow they ran and the version, yeah, I'm gonna believe it. And I would imagine that many other newer users you're trying to bring in are not even going to know what tools to run. They're going to be like running, was it NAPU? Is that how you say it? On Oxford Nanopore data. And I think like some people coming in to that are going to be like clinicians and just trying to get their feet wet. And they're probably going to look at that and say, oh, good, I have 60X. It's going to be good enough for NAPU. I'm going to run it. And they're not going to think too much about it. So I would say like me, I'm like extra cautious, always want to run extra QCs. But if these people say it's good and they can point me to how they ran it, I will believe it. But that's just me.
I think on the non-genomic side, I think similarly, because it's either I don't care enough that the rough of an expert group is going to be fine, or I care enough that no matter who did it or what they said, I'm going to have to do it again. And I think that applies to the genomics too. For finding the data sets, yeah, great, we got an estimate as estimate coverage, but for I have to do the specific thing, I have to know this, and no matter what, I have to go run my own pipeline because I need to measure something in a specific way because I'm doing something very niche. And so I feel like I think the same way, and I think, yeah, the provenance, how did you do this? Was it manually reviewed or that? Like once you've got that there, I don't think you can do any better than that. Yeah.
It feels a little bit about like the early days of gene annotation where they'd have these jamborees where 10 people would sit around and look at one gene model and debate every splice site. But I think, you know, the world we're in these days is like, I think people are becoming just more comfortable acknowledging that everything will have an error bar associated with it. But if we can capture the most common cases and maybe flag anything that looks exceptional, I think that would just elevate the use of all the data. Okay, so we have just a minute left here. I was wondering if anyone has any kind of final questions, thoughts, topics. We'll pick this up again in the town hall, but if anyone had any sort of final thoughts about data, now would be a great time to share. Yeah.
So I have a question about submitting data, and I get the option now when I go to submit to either submit to dbGaP or AnVIL, and I must confess I'm still afraid to click the AnVIL button, and I still click dbGaP. And I'm not sure why that is. I don't know if it's just like I know how the dbGaP process works and I don't know how the AnVIL process works or what, but I'd be like maybe interested to see like the advantage. Like if I put it in dbGaP, will it ever make it to AnVIL? Or if I put it in AnVIL, does it ever need to go to dbGaP? I don't know. It's a scary situation for me. Clicking the button is hard.
Well, let me make an important introduction, and I think she knows who I'm walking toward. Our master and wizard of data submission, Kate.
Yes. So yes, would like to encourage you to come to AnVIL. You can use Duos for registration as well. But yes, if you submit it to AnVIL, the dbGaP registration page will essentially direct them to AnVIL to find the data, but people can still submit their data access requests through dbGaP, and they will be able to access the data in AnVIL. With regards to onboarding, we have now extensive documentation to walk users through that, including videos developed by Allie, who I don't know if she's here right now. She may be in the other room, but then we also really are, we have a ticketing system and workflows, and we're moving towards self-service data ingestion as well to really guide you through the data submission process. So yes.
And the short version is I think we're like, I don't know, effectively functionally equivalent to dbGaP, but there's all this extra benefit about making the data accessible and having it in a place where it can be computed on and indexed and searched. And so, but if you ever have questions or feel stuck, just call me up. You're exactly the power user that we're trying to support here. Okay, thanks everyone. I propose we take just a 2-minute break. We'll regroup and then we'll kick it off for the town hall. Thanks everyone. Okay, well, welcome back. Welcome back, everyone. I think so. We're going to do a quick debriefing on how the breakout sessions went, and then we're going to kind of transition into the sort of open forum. As a consequence, like if there's questions along the way, just shout them out. Right? We have plenty of time here to kind of talk about this. So I'm going to go on behalf of sort of team data that was in this room, and then I see team tools in the background, kind of getting notes organized. So I'll try to give you a few minutes here. So in team data, we talked about several different topics. Some were more technical, some were kind of more procedural, some were more policy maybe. So it started kind of nuts and bolts about the virtues and maybe the opportunities for DOIs. I think there's a lot of excitement from Benedict's presentation that Docstor now automatically generates these and mints these. And I think there was sort of interest to kind of do this more broadly at sort of different data sets, potentially workspaces and other sort of artifacts. I think it may have even been Benedict that last time I met him saying a PDF should not be the only component to the scientific record. I mean, a PDF is useful. It does capture a lot of information, but often the PDF is just like one little sliver of all the sort of outcomes from a project. So I think we should embrace that. We should support that, you know, kind of more broadly. I know there have been experiments with certain journals about, you know, making workflows or containers or other sort of systems available. I think we should try to lean into that and be a leader in that space. So. Another sort of topic that came up was sort of acknowledging the fact that as much as we love AnVIL, there's going to be data sets outside of AnVIL that we want to sort of integrate and work on. At the top of the list were like access and interoperability with dbGaP through the National NIH Cloud Platform Interoperability Project, NCPI. There's a lot of discussions about at the technical level about what sort of APIs and standards are needed for this. For those of you in the room, just so we're all at the same page, there's work towards deploying something called RAS, which is a single sign-on technology where you could sign on to one platform and then you'd be able to kind of easily move data around in terms of knowing the system would automatically know which data sets you have authorization to use. So I like to think we're in the home stretch of getting all of the kind of sign off on getting these moving forward. So I'd like to think in the next year or so that will be kind of broadly available, and that should really help because then suddenly your dbGaP credentials will go with you no matter which platform you're on. In addition to RAS, there's a technology called DIRS. This is the registry service, the data registry service that will kind of present a generic URL where it doesn't matter which cloud, which platform the data set's on. It'll be a way where you can kind of easily move them across platforms. And there's been a lot of movement there to get DIRS support across the major cloud platforms. So I think at a technical level, these technologies are maturing. We talk all the time to our friends at dbGaP and RSRA. So hopefully, again, in the coming months and years, those opportunities will grow to make it even easier to integrate with those types of data. Another kind of big discussion topic was on like data harmonization, and this covered kind of a broad range of topics. One was there was an interest from the community about just like a variety of metrics being sort of presented in a summarized way. Some of these are like quite basic, like what's the depth of coverage on a data set? As we heard from, I think it was like Anshul's presentation yesterday, like data quality really matters. And if it's low coverage, low quality, then that can, there can be all kinds of, I call these dragons lurking in there that can really confuse the analysis. So one idea, one topic that came up is just like, let's either automatically compute those or record those on data submission. Maybe we would have like a modest budget, like $1 per sample where we go and sort of collect these. So it started with some of those basic metrics. There was some interest to have, more robust reporting on sort of metadata. A concern that was raised is if you have, very extensive requirements for data submission, that there's, 10,000 fields that need to be populated for every data set, that could become a barrier over time about slowing down the submission process and maybe even people getting frustrated and not submitting to AnVIL. So the kind of consensus is whenever possible, if we can do this in an automated way, those are definitely the metrics that we want to collect very broadly, even if they're maybe not 100% accurate. Even if there is a little bit of an error bar there, having automated metrics computed and available so that you can do these easy queries. There seems to be sort of great interest on this. We talked about, well, how could we, finalize the list of metrics to collect? The sort of agreement, the consensus in the room was at least that we just, we have enough expertise locally that we could draft such a list. And then, maybe get some community input to see if there's no blind spots. But this is a place where it was thought that AnVIL should sort of take leadership on sort of the metrics that were collected. And then sort of the last topic that came up was, I think some of this is awareness and sort of procedures and policies. As researchers are trying to decide, should we submit directly to AnVIL? Should we go to dbGaP or maybe other institutional resources or beyond? I think dbGaP has like sort of a known process, a known prior practice, some inertia. So I think on the AnVIL side, we're eager to talk to others to bring in their data. And, we like to think we have a lot of documentation, but I do acknowledge it can be confusing and overwhelming at times to know even how to get started. So what I'm hearing is maybe we should have I don't know, a webinar or office hours or another sort of set of tutorials that kind of walk people through this. I think there's something magical when you're like watching someone do this. It sort of just demystifies it. That's what I've really liked, for example, about the CoFest here, about just being able to watch Vivek train the model gave me the courage that it took for me to train my own model, for example. So I think that's a good model, and we look forward to that. I guess another kind of key takeaway is this meeting has been great, so that people get to know each other, and if there's questions that come up, we're trying to make it. We're just trying to optimize communication so that people know who to talk to as questions do arise. I don't know. So I'm here just on behalf of Team Data. Was there any other kind of questions, comments, thoughts that came up that we'd like to get recorded into the notes? I thought it was a nice sort of lively discussion. Okay, if not, I think we're going to transition to Team Tools and interoperability. Do we have a spokesperson? Okay, well, come up. Woo! Come up here, Fred. Having our notes. Oh, and please use the mic. This one, either. We're going to the podium and got this one. Okay, thanks.
Guess for Zoom, if the camera's there. Yeah, so we had a pretty nice discussion on 3 different topics. One is how to convey results to analysts, especially on the visualization front, what it means to be multi-cloud, and then what are the challenges for onboarding analysts that are comfortable with HPC but are new to working on the cloud. So for the first point on conveying results to analysts, I think this has come up in many of the tech calls over time. But basically, AnVIL, is not just a data deposition platform. It's not just an analyst platform, but it's also for people who want to interpret the results that are coming out of it. And I think Jupyter right now is probably, like the best way because, you can preview a rendered notebook. But if you want to get something a little bit more interactive, one of the things that's come up over time is Shiny. And, having a couple developers here, especially the ones that are very familiar with Kubernetes, there's discussion, again, just about trying to push the platform to support developers bringing their own Helm charts, right? And, not making light of the security considerations of that, but that would really enable a lot of developers who know how to develop this to bring it to platform and allow people to do it. Shiny apps just being one of the examples that Bioconductor is already doing. One question came up, I guess, about how Secret does this, because I think that's one of the examples of a more consumer side of viewing the results. But I don't think any of us really understood how Secret deployment development is happening there, but that was just one of the examples. There was a question about multi-cloud analysis for interoperability. And, aside from the egress costs and, appreciating that, I guess it's very non-transparent on the Microsoft side on what egress costs are. There was even the question of just how do you access data that is AnVIL hosted versus bring your own? And so Durst being the way that, there's this resolution to figure out what cloud it's on. But if you're bringing your own data and you're trying to share with someone else, is there actually some kind of service that helps people resolve that? Or is that up to the people bringing their data to advertise the correct way to access that data, whether it's on an S3 bucket or if it's something else? So I think maybe that's just documentation, or maybe there actually is some infrastructure needed for that. And then finally, I think, this is a very important thing because, from the outreach standpoint, there's a big push on, what audiences are we trying to train? And I think, one of the biggest people that we really need to make sure we're addressing are the analysts who are super comfortable on local on-prem HPC. What are the barriers to them adopting AnVIL as a platform? And there are parallels between, how do you do an analysis on Galaxy? How do you do an analysis on Terra? And just how do you do containers and things on the cloud? And I think a very specific use case that's coming up is, when people want to do a one-off analysis, like here's a new data set. I already know how to do this on my on-prem cluster. It already has all the packages, the libraries, like VCF tools being one of them. How do you sort of enable those analysts to be able to do that one step? And one parallel that was coming up was the way the Galaxy does it is that, there's 10,000 tools in the tool shed. So rather than being an entire workflow, which is hard to develop and debug, focusing it purely on here's the one step I want to do, which has, downstream making it a little bit easier on the development of, thinner Docker files, less dependencies, and trying to just say, okay, I've got that part one working. Now what's the next tool I need to do the next step? So maybe that being a model for getting analysts more comfortable with all the new technology of working with cloud, and different models and different things that need to go on there. There's an interesting thing about trying to do interactive environments. And I guess Bioconductor had this way in the past of having a Shiny app where you could sort of check boxes of what packages you want installed, and then it would assemble that container for you. So trying to think about if interactive environments had a way of sort of selecting, these are the things I want, and then making it easier so that people don't have to create their own custom Docker file, just some kind of service for that. And those are, I think, the things we have there.
Okay, so thank you for those great discussions and breakouts.